Hi everyone, we're here for a video on translation, and translation is going to happen after transcription. So we have our mRNA sequence here. I took those beads that I used in the last video and then I put them on a pipe cleaner so that it was a little bit easier to move them around as a whole unit. And so if we look at our mRNA sequence here, I've written it out and we can see we match up with an A, U, G, 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 C, A, U, U, and so on. Okay, so this mRNA sequence, if we put them all together, would line up bead for bead with the letters that they represent. And again, here's our key if you want that. So that's what we've got. We've got mRNA here, and then we have a ribosome being represented by the fabric. And so a ribosome is an organelle, and translation happens in the ribosome. So we're going to have any activity happening in the ribosome here. And then the whole table is sort of representing our cytoplasm of the cell. And so we have random tRNAs floating around, and those tRNAs have an anticodon on them, and we're going to see that later matching up with our codons. And then they have an amino acid attached to them also. So we see a variety of tRNAs around the table, and each tRNA will have an anticodon on one end, and then on the other end they'll have an amino acid represented by the circle with the shorthand letters on them. Okay. So for translation, what we want to do is we want to create a protein. That's the whole point. So we are going to move from mRNA, and from mRNA in the end, we're going to have a chain of amino acids, which is our protein. So to start, we're going to take our mRNA code here, and if you look across, you can see how we transcribed this, C's with G's, T's to A's, and A's to U's all the way down. And codons are in sets of 3. So at this point, we're going to take it and divide it by 3. So I'm going to count 1, 2, 3 nucleotides and draw a line. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, so now I've divided my mRNA, into codons. Now I'm going to do the same thing with my beads, just so it's easier for me to visualize. So I'm going to take the first 3 beads and pull them out to be their own codon, and then the next 3 and just leave a little gap. 3, 3, and 3. So we've got our beads separated into their codons as well. Okay, so for translation to happen, we're in the ribosome. The mRNA would in through the cell, make its way here. I'm actually going to put it at the bottom. And then what we need is for the tRNAs that are just randomly floating around the cell, right? Remember, it's aqueous. It's a watery solution in there. So they would make their way into the ribosome, and they're going to sort of come into this position of the ribosome. And they're going to see if their anticodon matches up with the codon here, okay? So the orange again represents A, so we have AUG, and that would pair up with UAC. So this is correct, and that would just hang out here. Okay, so then the next one we can see is GGC. And so perhaps, let's say this one came in, and GGC would need the anticodon of CCG, and this is not that anticodon. It's not correct. So even if it tried to get in here, it just wouldn't work, and it would float off and go elsewhere, okay? But we are looking now for CCG, and CCG happens to be on this tRNA down here. So this tRNA would come in, it would pair up, again, GGC codon with our CCG anticodon, and so these two tRNAs are right next to each other. Now at this point in translation, our amino acids need to become attached, and what I wanted to point out is the direction in which they attach to one another, okay? The methionine, MET here, this particular amino acid, is going to move over here and attach to glycine, okay? It will not go the other direction, because the next step is that this now empty tRNA is going to leave and float off into the cytoplasm to go attach to another methionine and ultimately come back and just repeat the process. This is sort of like a taxi cab, right? It goes and gets the one thing and brings it back, and the one thing and brings it back. So if we had gone the other direction, the whole thing would have floated away and we wouldn't be building. So methionine will move from this first position over to this amino acid, which remains attached. This one goes off and floats away to go find its methionine, and now we're here. Now we move to the third. So now we have AUU, which is what we have here, AUU. And so the anticodon to that would be UAA. And we can look around, we see UAA here. It would come in, it would pair up, again, A to U, A to U, A to U. And now, again, these are separate to start. Okay, these two are attached, this one's separate. And again, we repeat the process where now the glycine with the methionine still attached is going to move this direction into the isoleucine. And now again, our now empty tRNA is going to float away. Now, technically in our ribosome, this whole contraption, this whole set of connections here will be moving through the ribosome, or you could think of the ribosome as moving across the mRNA as we go, just simply with the sizes that we're working with for the benefits of the video. It's not exactly to scale, and so we don't see that movement. But this same process would repeat, right? UUG will mean AAC. There it is. And again, the cell is doing this on their own. There isn't something seeking it out. These are just floating around and happening to come in and match correctly. Matches correctly, our whole amino acid chain connects. Float away. Then we're on to our last two. So AUA, we're looking for UAU. Here it is. UAU comes here. Everything moves to the right. tRNA floats away. And then we need, or we have GUA, and so we need CAU. CAU is here, it comes in, separate these a little bit so we can see that connection, right? We've got our chain that's been built, we have our connection here, we see our amino acid chain pop over here, and then this one floats away. Now, some of them, like this one, are just not going to be used for this particular protein. It won't have any histidine in it. That's fine. Not every protein has every amino acid, so that's okay. When we get to, we would get a stop codon, and that stop codon, there's no tRNA that matches that. So at that point, we detach, and your amino acid chain would remain attached to each other. There's bonds connecting this, peptide bonds, and then this whole thing would curl up and form a protein. And now this is a protein. Perhaps it's hemoglobin, perhaps it's albumin, maybe it's keratin, right? Any sort of protein has now been formed by this process. And so it goes off, it separates, and it goes off to do its protein job, whatever that happens to be. This goes off again, in this case, to find another valine and get that amino acid and bring it back. Our mRNA strand now could go to a different ribosome and be translated yet again. So it's possible actually, because the ribosomes sort of move down the length of the mRNA, it would be possible for a ribosome to be working here, and as it gets about halfway through, another ribosome attaches and starts working. And so they just kind of at intervals start, and you have the same protein being mass produced in a really short amount of time. So our mRNA is translated over and over and over again and eventually broken down. The ribosome, again, would just go off to translate something else. That's what it does. So that's what it's going to do. And everything else is more or less recycled. Your body's very good at recycling different components. Now, if in this particular example, we had the tRNAs with the amino acids attached, and I was finding them on the table. For you doing this at home, the on-paper version, if you will, was going to use the book, and we're going to use our card here. So we have the mRNA here, and mRNA, folks, we'll put a little star here. The mRNA codon is what you're going to use in this table. So for our particular book, it's figure 10.9, but many of these exist. You can even just Google it and it will appear. So this is the genetic code. And what we see in the genetic code is you'll see a codon here. This one I'm pointing to is CUC. And that codon tells you which amino acid is coded for, in this case, leucine. Okay, but also CUA and CUG and CUU. All four of those codons code for leucine. Now again, the codon is what was on the mRNA. So when you think of this table, think back to the beads here. It's not the tRNA, it's the codon on the mRNA. What did we see here? And that's what we match up, and that's what we write down. So if we're going to do this with pen and paper, if you will, we would say AUG. Okay, so I know my codon is AUG. Back to the table. I see A's and UG. Now this one's highlighted because it is the only start codon we have. It's the only start codon every living thing ever uses, which is really sweet and evidence for evolution. But our point for this is that AUG is our start codon and it codes for methionine. So to write the answer of which amino acid to use, I'm going to use that shorthand of MET. That's my first amino acid. Because remember, we're making an amino acid chain. And I might put a dash here or a comma perhaps to just separate it. And then I'd go look for the next one. I'd look for the next codon of GGC. So back to the table. My first letter of the codon is here, G. My second letter is in this column, so I need GC. There it is, which is for glycine. So then I would write down the GLY shorthand that this book or table provides for you. And you would just continue that process through all of those. And what you'll see is as you take this codon, compare it to the table, you'll end up with the same amino acid chain that you and I just created here on the table, right, where we got in that same order. So technically, here's the answer to our practice video. So as you go through, this should be the amino acid chain that you would build from the table in this case to match it up. Now, final note, again, the tRNA carried the amino acid to the ribosome, and this anti-codon on the tRNA paired up with the mRNA codon, but the codon is what you use in the table. The tRNA is great, the anti-codon's great, but we don't use this in the table. We use the codon in the table. Okay, so that's translation, and that's what you get in the end. Amino acid chain, which is a protein, which is super exciting because you need those to live. So that's translation. Good luck. Have fun.